EMSL Summer School. Thank you so much for joining us again today. We're really excited for today's topic, which is going to be about natural organic matter. So I'll hand it off to Will now to kind of give us a summary of today's agenda.
Welcome, everybody. Today, we're going to be learning about organic matter characterization, so molecular level complex mixture analysis. And this morning, we got a plenary talk from Rene Boiteau, who's a assistant professor in Oregon State University. And then after that, I will be giving a talk about the characterization of organic matter using mass spectrometry. And Yuri Carrillo will give a talk about data analysis workflows. And then in the afternoon, we'll have some more workshops.
Great, thanks, Will. So we'll hand it off to you, Rene.
Great, thanks a lot. And it's good to see so many participants in this. So my name is Rene Boiteau. I'm an assistant professor at Oregon State in Earth, Ocean, Atmospheric Sciences. And prior to this, I was a postdoctoral fellow in EMSL. And so that was a really amazing opportunity to really start to get to know lots of different types of analyses that can be applied to study organic matter in the environment and soils in particular. So today I'm going to talk to you guys about some of the strategies that we can use to address knowledge gaps in understanding soil organic matter and some of the nuts and bolts of different analytical tools that can be used to address different questions, and then how you can apply those with different experimental designs. And finally, we'll go over a couple of case studies on specific experimental designs targeted to address specific questions and discuss a little bit how different tools can be effectively utilized together to provide more robust conclusions than one tool alone. So while I'm talking, if you guys have any questions, please feel free to post in the chat or on Discord, and I'll be happy to answer them as we go along. Okay, so to start with, I think it's useful to give a little bit of background about soil organic carbon and the carbon cycle, mostly just to get on the same page in terms of language we use to describe soil organic matter. Right? And so soil organic matter is in some ways very challenging to study in soils because it doesn't always mean the same thing. Soils are very, very different across the world, and soils can have very different amounts and compositions of organic matter. Right? But this might be the composition of a soil. This pie chart here might represent a fairly typical agricultural soil, where about 5% of the soil might be organic matter. Most of the soil might be some sort of mineral, and that might be sand, silt, clay, different minerals of different size. And then, of course, we have water and air that can be a very, very variable part of that soil as well, and of course temporally different. And so, you know, this slice of organic matter usually is a fairly minor fraction of the total soil, although in some cases it must be much larger. And ultimately, all of that organic matter is derived from plants, right? These are the dominant primary producers in soils. And so plants are fixing CO2 and incorporating most of that carbon into biomass as either leaf litter or roots, and then some small percentage might also be released into the soil more directly as root exudates. And so that input of carbon, organic carbon, into soils gives a very defined composition that depends on the plant that's growing and producing it, right? And then that organic matter is rapidly transformed by a lot of different processes, and most of those processes involve microbes, either bacteria or fungi that are living in the soil and using that organic matter as a carbon source, a nutrient source, and an energy source. And those microbes are really, really important for changing the composition of the organic matter, respiring it back into CO2, re-releasing nutrients, and also turning it into forms that end up being preserved over longer periods of time, right? So those microbes are chomping on the organic matter. Some of that organic matter gets incorporated into new biomolecules. Others end up as partially decomposed material that can accumulate. Okay, and so when we're talking about what is the composition of soil organic matter, I find it useful to divide it up into two different pools, especially when we're trying to understand how you analyze soil organic matter. So we have structurally defined molecules. These are either parts of living plants and microbes, and they might also be recently dead fresh organic residues. And so both of those have in common the fact that they have some defined chemical composition, because organisms are producing them through proteins that are templated in genomes, right? So all of these molecules can ultimately be traced back to some gene in the organism that made it, and some function associated with the production of that molecule. And then the rest of the organic matter is very polydispersed material that doesn't necessarily have a well-defined chemical composition, because it's the degradation products of microbial metabolisms, right? So this is an ultra-complex mixture of molecules that don't have repeated molecular structure, right? So some large polymer might be degraded through different pathways to produce a snowflake molecule that occurs once in some unit of soil. And there are many of these different molecules with very different structures that are all mixed together in some volume of soil. Okay, so if we look at example soil composition, a fairly small fraction of soil organic matter is usually these living organisms, and then maybe a slightly larger fraction would be fresh residues. Most of the organic matter that accumulates in soils is this decomposed material. Okay, so it's useful to think about, when we're thinking about soils, whether we expect to find our type of organic matter of interest in the structurally defined pool or this polydispersed pool. In practice, when we're thinking about the amount of soil organic matter and the quality of that soil organic matter, we need to come up with some sort of operational ways to define it based on different extraction procedures and analyses. Okay, so I mentioned briefly that all of the soil organic matter starts out as plant material. And so it's useful to kind of back up and think about what are plants made of, right? So plants and then the microbes that are also creating biomass from that plant material start out as different types of organic matter that have very different chemical structures and properties, right? So we have sugars and polysaccharides. Many of these polysaccharides are polymers that are used to build cell walls and structural material. Plants are also producing lignin, which is another structural polymer that's designed to be hardy and not broken down easily. Plants and microbes are producing a lot of proteins and peptides that are carrying out enzymatic functions. They produce lipids and waxes. So these are materials that are used to build membranes to protect cells from incoming materials. They can also include different types of signaling molecules, and then waxes that protect tissue from losing moisture. Then we have, of course, nucleotides, DNA and RNA that are carrying genetic material. Organic acids, these are often used as buffers inside of the cell, and then they can also be released to help modify the mineral environment and the chemical properties of the external environment in soils. We have pigments that are harvesting sunlight and have strong reactivities. And then this is a small subset of the different chemical groups that you can start dividing things into that include other metabolites like phytochemicals, osmolytes, and other things. So if we look at plant material itself, most of the carbon that is being taken up and fixed by plants that doesn't get re-respired ends up as sort of these structural materials, cellulose, hemicellulose, and lignin. So ultimately, a large fraction of the organic matter in soil ends up being transformations of these materials by microbes into lots and lots of other substrates. Okay, so let's think a little bit now about how soil organic matter degrades. So this is a net oxidative process, right? So we're going from some organic molecule. Here I have very simplified representation of this process. This might be the chemical stoichiometry of a sugar, CH2O, and that carbon is being oxidized. Some of it will get oxidized back into CO2, but the idea is that we're losing electrons from the carbon. Right? I say net oxidative process here because a lot of chemical reactions can end up reducing some organic carbon while oxidizing others. Okay, so as I mentioned before, this is largely operating on biopolymers that have to be broken down into smaller molecules that can be metabolized by the microbes who are carrying out most of these reactions. Right? And there are many different pathways that microbes use to break down complex materials like plant roots and leaf litter and other things that are being added to soils. Right? And those pathways depend on a couple of key things. One is the type of microbes that are present. Different microbes have different enzymes that can break down different materials different ways. We have different electron acceptors, right? So if you're net oxidizing the carbon, you have to be reducing something else, right? And so many of you are probably familiar with this redox ladder, right? So depending on the environment, different microbes can break down organic carbon with oxygen. When oxygen runs out, nitrate can be used. When that runs out, manganese can be used, iron can be used, sulfate can be used, and in the absence of any other options, then methanogenesis breaks down the carbon into methane and CO2. Right? And so different microbes are carrying out each of these degradation pathways, and the byproducts can be distinct depending on what microbe is carrying out what process. Right? So the point here is mostly that the degradation of organic matter is actually very, very complicated, and most of these reactions are very poorly constrained. So as analytical chemists and soil scientists, we have a very exciting job these days because there are just a lot of new windows into these processes that have barely scratched the surface of organic matter transformations and really linking those transformations to the microbes that are conducting the processes. And the promise of these investigations is that we'll be able to develop better understanding and knowledge of soils in a way that can help us predict changes to soil organic carbon stocks and also help us manage soil organic carbon in a way that can either help sequester CO2, provide better nutrition for plants, or stabilize soils. Okay, so then we have now covered why soil organic matter starts to break down from plant material and produce lots of different byproducts with different chemistries, both microbial, well-defined products, as well as these polydispersed materials. Right? So why does soil organic matter actually accumulate in soils? And as I mentioned, most of the organic matter that accumulates is this chemical breakdown byproduct. And so you can kind of think about preservation in three different modes. So one reason why organic matter may accumulate is recalcitrance. So the chemistry of that organic matter might be difficult for enzymes to access sites that can be pulled apart, or those chemical bonds might just be very strong, right? So there is a large energy investment in or steric hindrance to be able to pull that molecule apart, right? So this is something that is endogenous to the composition of the organic matter itself. But back in the day, decades ago, recalcitrance was really thought to be the dominant process, reason for soil organic matter preservation. But as we've become more adept at chemical characterizations, it becomes really clear that materials that could really easily be broken down can end up being preserved for quite long periods of time in soils. And so the reason for that tends to be these other mechanisms. One is adsorption, so either adsorption or some sort of attachment of material that is otherwise soluble and easily broken down to a surface where it is protected, right? So adsorption is the partitioning of soluble material onto particle surfaces through a variety of different types of chemical interactions. And then the final mechanism, which is somewhat related to sorption, is physical separations. So occlusion by particles or aggregates. So in this case, organic matter can basically be encapsulated by solids that prevent enzymes and microbes from accessing that material, so or redox elements. So instead of being broken down, it ends up being protected. And if that physical aggregation is broken apart, then that labile material could be accessed. Just like in the case of sorption, if there's a process that can desorb the carbon, then that material would be broken down. Whereas in the case of recalcitrance, that material is just stable. Okay, and so if we think about the relative importance of these different mechanisms, it really depends on environmental conditions. So this is a figure from a fairly recent review paper by Mike Rowley, and I really love this figure because it shows, one of the main knobs that control processes in soil is, of course, pH, and this represents different types of mechanisms that can preserve soil organic matter, and gives us a picture of the fact that different mechanisms can be predominant depending on soil mineralogy and pH. So in the case of the soils with lower pHs, they tend to be dominated by aluminum and iron-bearing minerals. And so the types of mechanisms that preserve soil organic matter reflect that, right? It's absorption to these clay minerals and reactive iron surfaces. In the case of more alkaline soils, those soils tend to have a greater composition of carbonates, calcium carbonates. And so then the mechanisms for preserving soils end up reflecting that. It's the flocculation of soil materials by calcium, it's the absorption to calcium carbonate bearing minerals. And of course, the nature of those interactions is a really important control on the standing stocks of carbon in soils. Okay, so as I mentioned, there are still lots and lots of different ways to add value to our understanding of soil organic matter. And there was a review paper that came out a little while ago from a working group that was thinking about, how do we model soil organic matter and what are some of the deficiencies in our understanding. And I pulled this table from that paper because I think, today still a lot of these details have not been worked out. And these give us some examples of the ways in which understanding of the mechanisms and the molecular scale processes occurring in soils can help provide us with better constraints for how soil organic matter pools accumulate and change over time. Right, so we can go through a couple of examples here, right? So insight into molecular structure is something that we're still just barely getting in our hands around, right? And most models today have decay rates of organic matter that are key to substrates and then modified just by moisture and temperature, right? But a more advanced and probably accurate way of doing this would be able to model decay rates as a function of substrate properties and positions in different environments, interactions with the specific microbial compositions, and a more complex suite of environmental conditions. So another example, humic substances. These are, this is an operationally defined term that is targeting that polydisperse material that comprises probably the majority of soil organic matter, right? So that heavily degraded material, right, has some intrinsic recalcitrance, resistance to breakdown, right? And so typically models have a cascade function that represents that material, right? But in reality, this material may be cycling very heavily by microbes, and so it may be more adequate to somehow incorporate the fact that this material is being broken up. Some of it is being incorporated into microbes, and then they're producing new recalcitrant organic matter as that material decays. So it's not this simple, once the material forms, then there's the slow decay until it all disappears. Okay, some other more topical questions circle around, what is the effect of specific processes like fires in affecting the composition and the fate of organic matter in soils. Permafrost, as permafrost thaws, it's really important to understand what the fate of that will be. And then there are lots of questions related to the physical heterogeneity of soils and how do we incorporate that into models in a meaningful way, instead of try to simplify things when we know that these physical separations and microstructures are really important to the fate of organic matter. Okay, so when I sit down with my students to develop a science question to study and a experimental design to study soil organic matter, I go through this exercise. And so we'll go through this together in the next section of our talk. So how do we develop an experiment to study soil organic matter? Well, of course, this starts with a science question that we want to answer, right? And then we start looking at what analyses are suitable to answer that specific question. Once we understand the types of analyses we're going to do, we can figure out what the sort of samples we need are and how many. And then we can figure out how we prepare these samples for analyses. Great. And one of the things that I think is really important to recognize about this process is that usually it's iterative, right? So we don't just go through this list linearly. Oftentimes we start with a science question, we start thinking about the analyses that are able to address that, and maybe there are some limitations to the analyses that are available. So we either have to develop new analyses, which alters our science question, or we have to adjust our science question to become tractable, right? And the same for these different steps. Okay, so the point here is that designing an experiment relies on both an understanding of the scientific system of soil systems and an understanding of the strengths and details and limitations of the tools that we use to analyze soil organic carbon, right? Those things are intimately tied to our initial design of an experiment. Okay, so we're going to go through each of these steps and describe how we can effectively conduct each one. Right, and so developing a tractable science question. The really important thing to keep in mind here is that you want to be as specific as possible to have a question that you're going to be able to answer in the time frame of an experiment in the lab or a field site, a field season. Okay, so these are the three categories that I start asking myself questions in to start to build a question. So I take this sort of modular approach. So the first step is to define the drivers that you're interested in studying. Maybe I'm concerned about changes in temperature in soils. You know, as climate warms, many regions are going to experience temperature changes, and there are going to be a lot of effects that are important to understand there. Or, you know, aridity or levels of moisture being developed are going to be different in the future. And so this might be the main science driver that's motivating my research. It may be nutrient cycling. So, you know, as land use shifts, it's important to understand what makes different types of nutrients like nitrogen and phosphorus available or limiting to different to plant growth and microbial processes in soil. I might be interested in something like seasonality, or maybe something more episodic, right? So what are the effect of fires that are, you know, raging across many places in the world as, you know, climates change and different places that used to be wetter become drier? What are the effect of floods, etc. Okay, so, you know, one thing is understanding the drivers you're interested in looking at, and then the next thing is constraining the types of soils that you're interested in studying. So because soils are such a mixed grab bag of materials, right, it's really useful to try to constrain our experimental design to think about one type of soil environment, or maybe compare one or two or a couple of types of environments in very specific ways. So that, because the dominant processes might be very different in one environment than another. And so if we compare, you know, forests versus grasslands, you know, forests are these environments that the dominant producers are trees, you know, the ground is shaded, the interaction between the plants and animals is really different. The composition of the soil organic matter is different. The minerals are usually different than grasslands, which tend to be dominated by grasses, you know, different environments, more arid. different animals and microbial processes going on there, different mineralogy. Similarly, we might think about, if we're interested in studying some changes in moisture, that floodplains have very different processes than coastal environments, where we're being impacted by sea level change and tidal cycles. And then finally, we can start to narrow in on a more specific type of organic matter and mechanisms that we're interested in studying. And the goal of this part of developing a science question is to figure out how we're going to get the soil organic matter out of the soil in a form that we can analyze. And this is in many ways one of the most difficult parts of designing a soil organic matter experiment. Most of the material in soil is very insoluble. That's why it stays in soils. But most of the analyses that give us a detailed understanding of soils requires us to get the material into a solution phase. So there are different ways to do that, and they tend to target different types of organic matter. And so if we can ask questions that are suitable for different ways of obtaining that material, it will be more effective, right? So we might be interested in water-soluble organic matter because this is the material that can actually be transported and taken up by plants and microbes in soils. We might be interested in microbial or plant metabolites because these are functional molecules that are directly tied to microbial composition and the health of plants. We might be interested in mineral-bound materials because these tend to be preserved for long periods of time. Maybe we're interested in processes regarding nutrients or trace elements, either micronutrients like iron and zinc, or toxins like copper or cadmium or mercury or lead or uranium, plutonium. Maybe we're interested in something like pyrogenic organic matter if we're looking at fires and want to understand how organic matter has been transformed. So the more we can narrow to a specific question, the easier it will be to develop, the more likely to succeed our experimental design and ultimately our experiment will be. Okay, so we can go through a very brief example of developing a tractable science question. And I encourage anyone who is interested, maybe post in the chat some other examples of science questions that you're interested in answering. I think one of the easiest ways to learn about different ways to develop a science question is to go through different examples. And so here's an example of maybe what I might start with as a science question that really isn't specific enough. So how will global warming affect the abundance and composition of soil organic matter? And so this is definitely a very topical and important question, but it's not clear how to develop a science question around something so broad, right? And so, especially with real world constraints, where you're usually working in a lab group with some limitation of scope and some time constraints. Okay, so we might go through the three different areas, right? What are the drivers that we're specifically interested in looking at? And so maybe, looking at the IPCC report, we can recognize that, sort of the high end of global warming changes might be around 2 degrees Celsius. And so we might focus in on that temperature change. What types of soils are we interested in? Okay, so it's hard to sample everywhere, but boreal forests are one of the largest biomes, and maybe these are an environment that are fairly easily accessible to my research group. And so this is a really nice area to just focus in on and understand how they will be impacted by temperature changes. Right, and then what types of organic matter and mechanisms are we interested in studying? And so if we look at boreal forests, most of the organic matter that's preserved there is somehow bound to iron oxide mineral surfaces. And so if we can understand how temperature changes are going to affect that material, right, we'll have some fundamental insight into the effect of future global warming. So we can put these together as a more specific question. How will a 2-degree increase in temperature affect the abundance and composition of iron oxide associated organic matter in a boreal forest after 2 years? So the 2 years part might not be very realistic, but we also have some real world constraints where if I'm trying to finish up a project on a reasonable time scale, we need to set some limits there. And this would be a question that we can design an experiment around, get samples, and actually look at how these processes are occurring. So let's see if we got some other questions? Other?
Oh, we'll take questions at the end, Rene.
Okay. Okay. We'll just keep them at the end. Okay. Thanks. Okay. So that's sort of a summary of how I would suggest going about developing a scientific question. So what are the different analytical tools that we can use to study the composition of organic matter in ways that give us mechanistic or molecular scale insight into the processes, things that we can link back to specific types of microbes and plants and that are inhabiting the soil. So one of the commonly used techniques and one of my specialties and one of the topics of most of this section is mass spectrometry, and in particular, ultra high resolution mass spectrometry. So this would be ion cyclotron resonance, ICR mass spectrometry, or perhaps Orbitrap mass spectrometry. And so one of the challenges in studying organic matter in soils is that it's extremely heterogeneous, right? And that means that if you look at the mass spectrum of soil organic matter, there are many different molecules that have very, very similar but slightly different masses. And so an ultra high resolution instrument allows you to separate mass peaks of molecules that differ by just very, very tiny mass differences. Again, these are very, very common in soils. So without instruments like this, you basically just have one broad peak at every mass in a mass spectrum, and that doesn't give you any useful information. So the development of ultra high resolution mass spectrometry and the applications to soils was really a phenomenal breakthrough in the study of organic matter, because for the first time, we were able to detect individual molecules in soil and start to characterize them and compare them across different samples, different soils. Okay, so there are a couple of different flavors of ultra high resolution mass spectrometry. You can do direct infusion mass spectrometry, where you just take it and infuse it into the instrument and get one spectrum per soil. So this is very high throughput. You see a lot of material. It doesn't involve, it's less time intensive, and there are some trade-offs. On the other hand, it has very strong matrix effects that make quantitation very, very challenging. So this is a more qualitative analysis. You can also take that material and attach a liquid chromatography system to the ICRMS on the front end. And so this allows you to separate organic molecules, and it improves quantitation. It allows you to see a much larger diversity of molecules because it reduces ion suppression and matrix effects. And Will and Yuri will talk a little bit more about that later in the session. But the downside of that is that you are more limited to the range of organic molecules you can analyze within a single analysis, and it's also lower throughput. And so this would be a method that's used for broad characterization and perhaps untargeted characterization. On the other hand, there are targeted mass spectrometry techniques that use either gas chromatography or liquid chromatography to look for the abundance of some specific analyte. And so the advantage here is that you can be much more sensitive, and you can also get absolute abundances of the analytes of interest, right? So if you have specific molecules that you're interested in that are markers of different processes or representative of something coming from a certain plant or microbe, then it's always better to use targeted methods if you're trying to look at abundances. Okay, so in addition to mass spectrometry, different types of spectroscopy are oftentimes very, very useful for studying organic matter. So unlike mass spectrometry, most spectroscopy approaches are non-destructive. That means after you analyze a sample, you can still use that sample for some other characterization. They're often quantitative, which is often a challenge for mass spectrometry. And they're used for the analysis of specific types of organic functional groups. So usually you don't get down to the molecular level with spectroscopic techniques. In some cases, maybe you can with NMR, but for the most part, it's telling you how abundant are different types of functionalities within your organic matter. So the probably most useful of these methods in studying soil organic matter is NMR, nuclear magnetic resonance. So this can be conducted on liquid or solid phase samples, and it gives you detailed information on hydrogen, carbon, And in some cases, if there's enough material, you can also look at nitrogen and phosphorus bonding environments. So you can look at the functionalities of these different elements in samples and compare them across systems. The information, again, usually is a little bit, usually peaks in NMR from soils are quite broad. So you get broad classifications of different functional groups, not down to the molecular level. So this is particularly useful if you're expecting some pretty major differences in the composition of organic matter across the samples that you're comparing. If you're looking at more minor differences, they might not show up in NMR. Also, NMR is fairly low throughput compared to some other techniques. Infrared, Raman, and X-ray absorption spectroscopies are other options. So these are also have their own strengths and weaknesses. They're sensitive to a broad range of functional groups. And in some cases, some of these techniques can be surface sensitive, which can be really useful because you can actually study the organic matter that is on a solid instead of relying on the ability to get it into a liquid form. But usually those techniques have a trade-off where you don't get as much specificity or deep information on the functionality. Okay, and then we have other techniques like UV-Vis and fluorescence spectroscopy. So these are useful for different, for quantifying different types of functional groups. They can see transition metal complexes and certain types of organic molecules that have lots of conjugated bonds, things like fluorescent pigments and aromatic molecules, which are often present in fairly high concentrations in different soils. Okay, and then finally, the other class of analyses that's really useful to pair with the others is elemental analysis. This can include high temperature combustion elemental analysis, which allows you to quantify carbon, hydrogen, and in some cases, nitrogen, sulfur, and phosphorus, and perhaps even some halogens in soil organic carbon. So you can take a liquid sample and measure the absolute concentrations of all of these elements associated with carbon. And then for less abundant elements, we can turn to inductively coupled plasma mass spectrometry, ICP-MS. So this is used for the quantitative analysis of metals and other elements in soils. And in the case of ICP-MS, you can sometimes do speciation analysis as well. So if you're interested in the chemical forms of organic molecules that have sulfur or metals associated with them, you can do a separation on the front end and actually quantify the absolute concentration of those elements in different chemical forms, which is a quite novel and exciting field. Okay, so when we're thinking about what analyses are going to be useful for our own experiments, we have to recognize the strengths and weaknesses of the techniques. Okay, so I'm not going to go here into a lot of detail, but it's useful to, when we're doing this assessment, recognize we're looking at the differences that the techniques offer in terms of the level of molecular or structural detail, the breadth. So, are you seeing a small subset of the organic molecules in our sample? And what is that subset? Or are you looking more broadly? How quantitative are our results? So can we attribute differences in signal to differences in the abundance or relative abundance of something across samples, or is it more complicated than that? And then preservation of in situ conditions. So a lot of these techniques require sample preparation that can change the chemistry of molecules. And so it's just really important to recognize what potential changes could have occurred. And if you're trying to look at something that is in the more native conditions, that might exclude different chemical analyses. Right, so some examples, FTICRMS, high resolution, ultra high resolution mass spectrometry observes a pretty small subset of the molecules in our sample, right? We can only look at a specific mass range of molecules. We lose really low or really heavy molecules. And then it has poor quantitation. So the composition of our sample, the other things in our sample beyond what we're analyzing can affect sensitivity quite a bit. But on the other hand, it has very high sensitivity. So we can detect very low abundances of material, pretty high throughput, and then it provides molecular elemental stoichiometries, which is a really powerful aspect of FTICRMS. NMR might be in a counterpoint where it it looks at all of the carbon in your sample and can be quantitative. So you can tell how much of your carbon is associated with aromatic functional groups versus carboxylic acids versus aliphatic material. But it tends to have low sensitivity and throughput. And usually it doesn't resolve fully the molecules that are in soils, just because they're such a complicated mixture. So every method is offering us a different and incomplete window into organic matter composition, and we want to understand what those limitations are, and also the useful ways that different techniques can be used together. Okay, so once we have an understanding of the techniques that we want to use, we can start to develop a sample set. And I think that it's really useful to start thinking fundamentally about what are the specific comparisons we're going to make across our system. So there are lots and lots of uncertainty that arise from every decision we make about how we collect and preserve our sample. And most of these uncertainties can be dealt with if we have careful comparisons. And so we might have two different conditions that we're comparing against. Or maybe 3 or 4 different comparisons, depending on your experiment. Or we might do something like look at a time series, right? Okay, I have this one soil. What is going to happen over time if we change temperature or if we change soil moisture or some other properties? Okay, and then we can also design experiments in 2 different ways. One is to design a field campaign where we collect samples that we're going to analyze and compare, versus collecting samples that we're going to bring back to the lab and incubate to look at the effect of a specific change on soil carbon composition. Right, so field samples are representing real world conditions, right, but oftentimes those are very complex. And so incubations are useful for trying to elucidate specific effects and mechanisms. And so again, it's useful to recognize whether your science question is better suited for designing an experiment where you're just looking at field samples that are compared across 2 similar sites that differ in some key way, or whether you're going to collect one soil and incubate it in different ways to understand how that soil is affected by a specific process. Okay, so once we've decided on our samples, we have to start thinking about the organic matter pool that we're going to target. And I think we can pretty effectively break this down into sort of 3 different pools. So we have the roots and rhizosphere. So these are the plant components that are actually putting fresh organic carbon into soils and are hotspots of microbial activity. So we can pull out plants and then look at the root associated carbon or the roots themselves. We can take soils and we can analyze this material associated with the soil directly. And usually when we do this, we're separating that soil into some size fractions in some way. So we can use sieves to collect finer material or coarser material. And then finally, we can get pore waters, right? This is the water that contains material that ends up dissolving and absorbing dynamically in the soil environments. And this is actively turning over carbon that can be transported and taken up by plants and microbes. And so for many types of studies, that is of interest. And we can use rhizome samplers or something similar to obtain that sort of material. Okay. Other details of developing a sample set is deciding on how much replication we need across the different groups that we're trying to compare. This is really, really important to decide early on, and again, might involve some sort of iteration where you do some analyses to evaluate how reproducible a specific analysis is, and then use that information to figure out how many replicates you need. So soils are really, really heterogeneous, and the bottom line is that the more replication you have, the more subtle the differences are that you can elucidate, right? So if you don't have that many replicates, maybe you can see really broad changes, but if the changes are more subtle, then you might need higher replication to have more statistical significance. And so I think that's another really nice focus of this, this workshop. Okay, it's also useful when you're designing your experiment to recognize that these replicates and blanks add up really quickly. So keep that in mind when you're designing your experiment and do it from the very beginning. Okay, and then it's really important to have quality control checks for any chemical analysis, right? So you should always think about the quantification or model compound standards that are relevant to your question that you can analyze before and after a sample set to make sure that analytical conditions are consistent over time. Oftentimes we use pooled samples to evaluate quality control. So this would be taking a little bit of every single sample that you want to analyze and mixing them together and then running them at the beginning and then punctuated throughout your sample batch and at the end. So, you know, if there are any major deviations to the repeated analysis of that material, you know, you recognize it and you incorporate that into your interpretation of your data set. And then internal standards. These are things that you're going to spike into your sample and be able to detect as a way to evaluate how sensitivity or some other analytical criteria has been held constant or maybe deviated over the course of analyses. So having good quality control checks designed into your experiment saves you a lot of headaches later on, because if anything goes wrong, you know exactly what has happened and you can deal with that by either rerunning some samples very easily or just incorporating that information again into your analysis and conclusions. Okay, so this is just a brief example of sample set that I might come up with, right? So if I'm running a bunch of samples for mass spectrometry analysis, I might start with a water blank and then run a standard mix and then a process blank. So this is water that's gone through every single step of my sample processing. So I can figure out what is from contamination from my process versus what was in my samples. And then I might have 5 replicates of 2 different types of samples. And you might consider whether you want to run those in order or randomize them. And randomizing them can get rid of any effect of batch effects where instrumental conditions might change over a run. And then I rerun the process blanks, the standards, and the analytical blanks, so I can see whether they're the same or different at the end. Okay, so then preparing samples for analyses. Many of these analytical methods require getting organic matter out of the soil matrix and into solution. Oftentimes you need fairly high concentrations, so you need to somehow pre-concentrate that organic matter. And oftentimes you have to remove particles, salts, or other metals that can interfere with an analysis. Right, so it's important to understand how this sort of processing affects composition. Different types of chemical extractions are going to target different types of organic matter. And in many cases, different chemical reactions can occur during the processes that are altering composition. That's just the nature of any extraction. And so it's okay if this happens, but you should be aware of the types of reactions that can occur and how and make sure that they're not affecting your conclusions. Okay, so what characteristics of organic matter affect its ability to be extracted by different methods, right? So different organic molecules have different chemical structures, and that give them different solubilities or polarities. So very oxygen-rich molecules are very soluble in water. More aliphatic molecules, things with straight carbon chains, tend to be more soluble in organic solvents. Many organic molecules have acid-base properties, and so the pH at which you do an extraction has a huge effect on whether you get molecules that have acids or base functional groups out of solution. And oftentimes, you can target one or the other, or you need to do some sort of sequential extraction to get them both. Molecular weight can affect their solubility and analysis. Some molecules might be sensitive to redox, light, or microbes. In many cases, there are living microbes in our soils that can degrade things over time. So it's very useful to understand how microbial degradation can affect composition in your experiments. And then some molecules are volatile, right? So if you're having the samples sit around for a long time, you might lose them as they evaporate. So these are the categories of common approaches used to extract organic materials. It's not a comprehensive list, but there's this nice review paper that came out just last year that describes these different approaches in more detail. So I'll sort of leave it at that. The different methods are basically targeting different types of organic matter pools, and they can be effective for addressing different types of questions, right? So for example, water extractions are getting that soluble component. Alkaline extractions would be where you add a strong base, and that is really good for getting lots of organic carbon out of soils, but you're also breaking lots of bonds and changing the chemistry of the molecules in order to get it in solution. And so it's just really important to understand that what you get from that process is chemically distinct from what was there. Mineral dissolution, very similar, has these potential reactivity concerns. And then for more non-polar molecules, you might have to do organic solvent extractions. So this highlights, again, the reason why comparisons are really, really helpful, because if your questions are designed, again, around, how does this process affect composition, and you're analyzing two samples that have been through the same extraction procedure, a lot of the potential difference, the potential underlying differences can still be preserved, even though there's been some alteration due to extraction. And then finally, we have sample cleanup and preservation. So these are just useful tips. In general, we want to avoid long-term storage of samples, but usually there's going to be some sort of long-term storage. So if you have to, and again, this is very common, you can filter, sterilize, and freeze a sample. So the reason we do that is to avoid microbial degradation or further oxidation of the organic carbon. Oftentimes, we have to remove salts and other interfering agents in our samples. So for any type of mass spectrometry, this is very, 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 very important. And this ends up being done usually with a solid phase extraction. And there are lots of different choices about how you can solid phase extract something. So, the decision on how this is conducted really gets down to the question and what types of organic matter someone is interested in targeting. And then finally, there are some ways to concentrate organic matter. So solid phase extraction can do this, and then another way to do this would be to freeze dry your sample. So anything like that is going to, of course, introduce a loss of volatile compounds. So I think the main takeaway from all of this is that any decisions that you make about soil sample handling, the important thing is to be able to have some rationale behind your choices, right? No decision is going to be perfect in that it will get every single molecule that everyone is interested in. But as long as you can justify your choices and be consistent, you can develop an effective experiment. And then another very helpful tip is using very clean materials and solvents, right? So especially for mass spectrometry analysis, the kiss of death is if you sample and have plasticizers or some other organic contaminants that swamp all the signal of your organic matter. This is really, really common if you're not careful. So here are some tips to avoid that, right? Use very high quality solvents, avoid soap, try to avoid plastics in general, but if you need to use them, and oftentimes we do, use high quality clean plastics. And lots of us are able to help you decide on what constitutes clean plastics. And then again, just a reminder to prepare blanks and quality control samples. And those allow you to evaluate contamination and where that contamination is coming from. So usually there's going to be some sort of contamination in your sample. And the important thing is to be able to know what that contamination is and prevent it from affecting your ability to detect what's in your sample. So all of this sample prep advice is just step one of decisions that you have to make about how to analyze your samples. And there are similarly important questions to ask about your instrumental parameters, how you develop your analysis itself, how you're going to analyze and interpret your data, and then ultimately what you're going to do with your data. And the point here is that you just want to start thinking about all of these steps at the very beginning when you're designing your experiment, so that you know that by the end of it, you're going to have something that gives you a robust answer to your question. Okay, so now we'll briefly go through two case studies of how you can use mass spectrometry in particular to address specific questions about soil organic matter that have relevance to changing world. And so this is an example from a paper from a few years ago by Colin Ward, looking at permafrost, thawing permafrost in the Arctic. And so the question here was pretty simple. It's how and why does sunlight impact microbial respiration of dissolved organic carbon that's draining from permafrost soils? And so we know that as the Arctic thaws, permafrost end up draining lots of dissolved organic carbon, and most of that carbon is oxidized by two different processes, either sunlight or microbes. And the question they were trying to address here is, well, are those really independent processes, or does photochemical degradation actually affect the ability of microbes to further degrade organic carbon? Because they're not necessarily degrading the same things. And so the choice of analysis here was direct infusion ICRMS, so collecting dissolved organic carbon, and then looking at differences in composition across different conditions. So samples were selected from permafrost and organic layer soils that were extracted with water to get that what will eventually become dissolved organic carbon. And then sunlight was, they had a set of samples where sunlight was shined on them, or simulated some sunlight, and then samples that were kept in the dark. So that's one comparison. And then they took that carbon after incubating them in the light or dark, and then either added bacteria to them or did not. Okay, so this gave them a matrix of conditions that they were comparing across to understand how does carbon change under each condition. So from each of the samples, they were filtered and then solid phase extracted onto a hydrophobic resin that, again, is just capturing a portion of the organic carbon. So this is just giving you one window into the changes of organic carbon, but it's giving us a molecular window into those changes that helps us understand the connectivity between photodegradation and biodegradation. Okay, so they designed their experiment such that they were able to compare a couple of bulk rates across their sample, and that informed the effect of the different processes, while the chemical analysis gave them some insight into processes, into the mechanisms, right? So from their incubation experiments, they found that after permafrost layer dissolved organic carbon was irradiated with sunlight, so this plot is showing respiration rates, either O2 measured, CO2 measured, or microbial production and growth efficiency across different samples. And then the difference between light versus dark responses. And so for permafrost layer dissolved organic carbon, the light treated samples had much higher respiration rates than the dark treated samples. Whereas for the organic layer samples, there was a much greater, there was really no effect, perhaps a slightly negative effect on light on microbial respiration. Right, so looking at the composition of organic matter across the different samples, they found they were doing a pairwise comparison and looking at the differences in compounds that were either produced or degraded by photochemical processes or biological processes. So again, the nice thing about this experimental design is that they're staying with the same organic carbon material, the same dissolved organic carbon extract, and just looking at differences after irradiation or differences after biological activity. And so all of the general matrix of the sample, which can affect ICRMS analysis tremendously, is the same across all of their different treatments, all of their different comparisons. Okay, so they found a bunch of different molecules that were being produced by photochemical degradation. And this is pretty consistent with what other people have observed and confirmed with NMR, where when you shine light on organic carbon, you tend to break off carboxylic acids and things that can bind to metals, and your general organic carbon becomes less rich in oxygen. And so in general, many of those molecules overlapped with many of the molecules that were biodegraded overlapped with molecules that were photochemically produced. And so one of the conclusions of this study is that sunlight is converting some of the dissolved organic carbon into permafrost into compounds that could then ultimately be degraded by microbes. And so there does seem to be this interaction between microbes and sunlight in permafrost. And so what would be add value to a study like this is, I think, a targeted study looking at, how this process might occur with a specific compound, or pairing these types of analyses with something like NMR that can confirm some of the changes that they observe. And basically pairing it with a more quantitative technique. And then I'll briefly go over another case study, and this was from my own research, where I was looking at how metal deficiency affects the processes that solubilize minerals in soils when you have a mixed community. And so here I was interested in understanding, as land use changes, there are lots of different processes that have been recognized for a specific plant to dissolve minerals in their soil environment by producing organic acids, lots of different types of organic acids, or coumarins, or different types of organic molecules. But some of those organic molecules may accumulate in soils and serve that function. Others might be degraded by bacteria. And bacteria do the same thing. They have a suite of molecules that they can produce that can dissolve minerals and affect soil preservation, carbon preservation, and the availability of micronutrients. But again, other microbes can break down those compounds as well. And so the question I was trying to address here is, when you have a mixed community of a plant, a grass, and bacteria that each have their own strategies for dissolving minerals, how do those metabolisms interact? So it's really getting at fundamental mechanisms that can be tied back to biosynthetic pathways and transporters and genes. And so for this, I used liquid chromatography coupled with mass spectrometry as an analysis, and nuclear magnetic resonance as another one, to look at the composition of compounds being released by different plant microbe setups. And so the sample selection to address this mechanistic question was to simplify the system, actually even simpler than real soil, and grow things semi-hydroponically. So the soil matrix itself was just silica beads, a well-defined soil, and they were watered with a nutrient solution that either had iron in it or did not have iron in it. So we're comparing these plus or minus iron treatments, and then we had treatments with and without bacteria. And so we were able to look at how these different treatments affected plant growth. So just visually, you could see that the iron treatment affected plant growth significantly. When we didn't add iron, the plants were yellow, the leaves were yellowed, which is a sign of iron deficiency. And it's a very common issue in alkaline soils. When we added iron, the plants were greener. And if we look at biomass, there was a significant difference in the amount of biomass from the iron-treated plants versus the iron deficient plants. Right? And we expected this, right? When we look at the effect of bacteria, there was no statistically significant difference. So it didn't seem like the bacteria themselves really affected the growth of the plant. So these root exudates from these four different conditions were collected and simply pre-concentrated by freeze drying for NMR. And this is, again, nice, the simplified system made it so that we didn't have to do a solid phase extraction before analysis here. Okay, so we were able to take this, all of these samples were run in with 5 replicates. And so we were able to do statistical analyses that looked at what features, what molecules in our systems were different across our different treatments, which of those differences were statistically significant, and how, what was the magnitude of these differences. And so we were able to identify a bunch of different molecules that were different across our different samples. And we were able to confirm the identity of, so the initial statistical analysis was done using liquid chromatography mass spectrometry, which sees the broadest number of distinct molecules. We were able to confirm the identity of a lot of those molecules using NMR. And then we were able to look at differences between our treatments to come up with some new mechanisms of cycling. And so one of the main differences in our treatments was that in the iron deficient soils, the grasses produced a much higher concentration of these two organic acids, neogeneic acid and deoxyneogeneic acid, right? And those were both the most, two of the most statistically significant and largest changes in the root exudate composition that we observed. And we also found that the bacteria degraded both of these molecules. So the bacteria that we chose, Pseudomonas, is also capable of producing its own organic acids and molecules that can break down the soil, but we didn't see any of those in this particular experiment, which was pretty interesting. And so what appears to be the case is that when these bacteria are growing with the grass, they actually turn off their strategies for dissolving metals and actually start degrading the molecules that the grass is producing instead. And so ultimately, knowing these pathways allows us to look for the specific transporters and the decomposition pathways within the bacteria that we were studying. And knowledge of, once we have those genes identified, we can look more broadly in soil communities and see what bacteria are capable of taking up and breaking down these important plant compounds that are contributing to mineral dissolution and metal bioavailability in many types of soils. Okay, so just a quick summary of some of the key points. Soil organic matter contains both well-defined molecules and complex polydispersed molecules. The sample preparation and instrumentation that we use is really important. And it's important to take some time to develop an experimental comparison and preparation method that can address a specific question. And oftentimes this process of developing the question is iterative with your understanding and gaining understanding of how samples are processed and analyzed. And then if we can combine analytical methods, we can often get results that have more depth and robustness than if we just apply a single approach. So thanks to everyone for your time. I ran a little over, but I think there's a break after this, so I took advantage of that. And I'm happy to stay on and answer questions.
Thank you, Rene. That was really good overview of quite a lot of complicated science areas. So I think everyone really appreciated that content. We have a couple of questions that came in through the Discord. We have one from Chelsea Saint-Germain. She's asking, for the first case study, do you know what bacteria they inoculated with and why they decided to use those?
Yeah, so that was with a natural community, and they did do sequencing of that community. I don't remember the details of that, but the idea was, is sunlight making the permafrost dissolved organic carbon available to the microbes that are naturally occurring in the receiving waters of where the permafrost DOC is going. But that's a great question. Yeah, and of course, yeah, there are different ways to design the experiment to address more mechanistic or broader questions that have more environmental relevance.
Cool. Thank you, Rene. We also had a question from Sandra Liu asking, would it be possible to measure volatiles in soil? If you're interested in microbial volatiles, is the amount produced to be too low to be detected by the instruments, e.g. GCMS?
Yeah, that's also a really great question. So you can definitely analyze volatiles, but usually the approaches that you use have to be tailored to volatiles, and maybe even the specific volatiles that you're interested in. So something like formate is a very common component in soils. It has some water solubility, but if you try to dry your sample, you would lose that. So that's an example of a molecule that you could see with a water extraction and LCMS or GCMS analysis if you prepare your sample correctly. But there might be other volatile compounds that you wouldn't see with that type of organic acid water extraction. So it definitely depends. If you're specifically interested in volatiles, you would want to try to get a better sense of the specific types of volatiles you are looking at, and that would inform your approach.
Cool. Thank you. I don't see any other questions right now. Oh, here we go. Here's a question that just came in. Andrew asks, you mentioned a little bit about tracking an organic molecule to see its degradation. How would you know which molecule to label given you're working in a complex system, say like peat?
How would you know which molecule to label given you're working in a complex system? So that's a really great question as well. And I think that it would require you to ask a specific enough question that dictates the answer, I guess. So, you know, you would want to consider what type of molecule is having a specific impact based on the driver that you're looking at, if that makes sense.
I think that makes sense. And then another question just came in. Malak Tafali's paper mentioned they did not use the PPL cartridge for soil DOC purification. Is that possible?
Yeah, so that's a really great question as well. And it is certainly possible to not do a solid phase extraction. But the reason why it's a good idea is because it gets rid of salts that are often very abundant and affect your analysis substantially. So it depends on your experimental design and the types of comparisons you're making. But one of the risks of not doing solid phase extraction is that you're basically measuring like salinity. You know, if you have a sample that ends up being a little bit saltier than another one, those salts have a huge effect on what you see in your soils, and that will basically be the biggest driver of any difference. And of course, that has no relevance to the composition of organic carbon, which is what you want to know. So it's always a good idea to do solid phase extraction. Even in the example where I didn't do solid phase extraction, we were doing chromatography, which is basically like an online solid phase extraction. So I'm a huge fan of solid phase extraction.
Thanks for answering that.
But again, it depends on the question and the soils. You know, if there's not a lot of salts and very organic rich, maybe it's not necessary.
I'll add, not as a spoiler, but my talk, which will start in about 15 minutes or so, I have a couple of slides showing what happens when you do and don't do solid phase extraction on the same sample. And as Rene said, you really need to a lot of the time. I don't see any other questions right now. I'll throw you a quick, easy curveball one. You know, most research labs don't have as many instruments as you've named today. Do you have any top tips on how to get access to more instruments and more capabilities?
Yeah, I mean, maybe that's slightly a leading question, but you know, I think working with user facilities is a fantastic way to become exposed to many more types of instruments. And so obviously EMSL is one of the top that we have access to, very fortunately here. There are different ways to engage with the user facilities. You can write exploratory proposals. There are a bunch of different types of proposals that can be written. And usually the scientists at these facilities are very eager to engage with new people who have questions. And so I think the biggest thing is to recognize that everyone who has, especially at these facilities, who has the expertise in different types of analyses are not expecting you to come in knowing everything about how these analyses work or even knowing what's available. So there's a little bit of reassurance that, you know, if you're eager to learn, there are lots of people that are eager to teach you about what you can do and help you develop science questions. And you know, I'm always happy to answer emails and help direct you to people.
Excellent. Thank you very much, Rene.